start. It's after 1 by that clock and after 2 by that one, so regardless, it's time to begin. And I'd like to introduce Ben Garling from the London Math Society, who would say a few words about Spitalfields and the program.
Thank you. London Mathematical Society was founded in 1865 in University College London. It was intended first of all to be a society for University College London. Immediately it became a society for London, and almost immediately, i.e. within a year or so, it became the National Mathematical Society for the United Kingdom, and it's stayed like that ever since. Founded in 1865, it's one of the oldest mathematical societies in continuous existence. But of course, it had various precursors before the London Mathematical Society was set up. The most notable of this is the Spitalfields Mathematical Society, which flourished from about 1760 to 1830, and its real heyday was almost exactly 200 years ago, in the first decade of the 19th century. There were two essential features of Spitalfields meetings. They held meetings in this decade. The audience would be something like 300 people. The first basic requirement was that the meetings should be general. They should be for a general audience, and they should introduce ideas to all sorts of people who hadn't met them before. 20 years ago, Christopher Zeeman at the London Mathematical Society thought it was really important that mathematics should be disseminated in this way, and so the London Mathematical Society started its program of Spitalfields Days, which were intended, often at a meeting like this, where there are specialists, for the specialists to take their ties off and relax and explain to a more general audience what the subject's really all about. And so that's why we're here today. The second feature of the Spitalfields Society was that it had an extremely complicated and obligatory system of entertainment. That it was important that people should come, they should smoke their pipes according to a certain rule. That's gone out right out of the window. And then they should also enjoy a pint of beer or something like that. Well, this meeting doesn't quite run to that level, but let me remind you that when we finish at 5:30 this afternoon, there will be a party afterwards, which I hope everyone will enjoy.
Thank you so much.
What is Spitalfields? Spitalfields is an area of London in the northeast, and it's where the hospital was. Spitalfields means the fields by the hospital.
Thank you very much, and thank you to the London Math Society, too, for sponsoring us. That's great. I just wanted to take about 30 seconds from Pete's talk to explain the title. And someone emailed me wondering whether or not there was a typo. The answer is no, that Yggdrasil is actually from Norse mythology, and it stands for the world tree of life, actually. It's a giant ash tree, and it seemed very befitting for this meeting. And so I suggest and recommend that you Google Yggdrasil after the meeting and see all the interesting mythological creatures that live in the branches of this tree. Okay, great. Our first speaker is Pete Lockhart from Massey University, and he's all ready.
Sorry? Yeah, Spitalfields one probably.
Okay, great.
And thank you. It's great to be here. I hope to disseminate some biology. I'm a biologist. Hopefully it's the sort of talk that might be enjoyed with a glass of ale, and may at the same time be interesting and informative. All right, so I'm going to talk a little bit about the development of some ideas in the context of a problem that I became interested in as a PhD student. And the problem really concerned the early events of oxygenic photosynthesis, the formation of organelles. Oxygenic photosynthesis is a really fundamental process. It's thought that it's evolved just once. It's that process where light energy is used to split water, to produce energy, to fix carbon dioxide into sugars. And it's carried out in prokaryotes, and it's carried out in some eukaryotes. In eukaryotes where it's carried out, it's actually carried out in organelles. These are remnants of photosynthetic bacteria, remnants of, if we were to talk about photosynthetic bacteria that carried out oxygenic photosynthesis, we might generally term them cyanobacteria. And it's thought that remnants of of cyanobacteria are the result of chloroplasts. Now, the formation of chloroplasts is a very ancient phenomenon. There are fossils of red algae, eukaryotes that are thought to have contained chloroplasts, as far back as 1.2 billion years. There's certainly microfossils of cyanobacteria back almost a billion years, and then there's byproducts of what are thought to be oxygenic photosynthesis going back considerably earlier than that. So it's a very ancient process, and if we're interested in reconstructing the evolutionary relationships of organisms that undertook or carry out oxygenic photosynthesis, and we're interested in getting at those very early lineages, you can well imagine it's a difficult problem because we're talking about evolutionary relationships that are very ancient indeed. Many that we're interested in predate, 1.2 billion years. All right, so there are these, if we were to talk about the three domains of life, we might think of some supergroups, eukaryotes. There's at least 5 supergroups. And in 4 of these supergroups, these lineages, these are largely today defined by results from DNA sequence analyses, where people have reconstructed evolutionary trees and they see these major clades. And so we've got one group, the Opisthocont here, which has sort of animals in it, fungi. This amoeba group, a group that contains the plants, the seaweeds, the algae. There's a group called the Excavata, where we have funny euglenoid type organisms. Rhizaria, another peculiar group. And Chromoevolata, which is a group that contains also seaweeds, brown algae. Now, in 4 of these groups, there are organisms that carry out this oxygenic photosynthesis. In 4 of these eukaryotic groups, there are chloroplasts. And you find quite a diversity of chloroplasts or organisms with chloroplasts. And as I stand here today, there's still a lot unknown about the types of chloroplasts and the genomes of the organisms that contain these chloroplasts in many of these groups. In one group, we have the green plants, the plants we see around us, also the red algae, and another unusual group called Glaucophytes. I'm going to be talking about the relationship between these taxa somewhat. We have in the Excavata, we have organisms that contain green chloroplasts. Rhizaria, there's some unusual organisms that also contain green chloroplasts. In this group here, which contains the brown algae, we have brown chloroplasts, red chloroplasts, green chloroplasts. We also have some organisms such as malaria where you find remnants of oxygenic photosynthetic organisms, but these chloroplasts no longer carry out photosynthesis. They have some other present-day function. So the question of interest, the general question was, how many events of endosymbiosis have there been in the origins of chloroplasts? So endosymbiosis, this is the swallowing of an organism by another organism. And people talk of primary events of endosymbiosis. So that's a swallowing of a cyanobacteria-like prokaryote by a eukaryote. But then there seems to have been both secondary and tertiary events in some lineages. So that organism that swallowed a cyanobacteria itself was swallowed by another eukaryote. And then again, this has happened at least 3 times in some lineages. And biologists have been interested in trying to reconstruct the evolutionary relationships among the different chloroplast types, and also trying to understand the relationship of the evolution of the particular chloroplast types in these different eukaryotes with present-day free-living cyanobacteria. These are the bacteria that carry out this oxygenic photosynthesis. And what we find, if we look around for present-day cyanobacteria, we see they're characterized by different pigment types. There are some that, to keep it simple, there are some that have red pigments. These are phycobilly proteins, complexes. There are some prokaryotes that have chlorophyll A and chlorophyll B. You don't find examples of cyanobacteria that have both. So I've got red, green, I've got red, and I've also got yellow here. That actually denotes chlorophyll C. So if I was to talk about pigments, I would say red, phycobilly proteins, green, chlorophyll A, chlorophyll B, yellow, chlorophyll A, chlorophyll C. And we see these pigment types in various chloroplasts in different eukaryotes. And early on in the study of these organisms, there was a hypothesis of multiple events of endosymbiosis at the primary level. So what do I mean by that? Cyanobacteria with these red type of pigments gave rise to eukaryotes that had the red pigments. The cyanobacteria that had the green pigments, chlorophyll A, chlorophyll B, gave rise to all the higher plants we know, but then also some other chloroplasts and some other unusual eukaryotic lineages. Those with the brown, in actual fact, nobody's ever found a cyanobacterium with a chlorophyll C, but it was thought that such a cyanobacteria has given rise to those chloroplasts that contain chlorophyll C. Then, however, there was some DNA studies done about the time I began my PhD, and some trees that were reconstructed to investigate the evolutionary relationships suggested a different hypothesis, that there was perhaps one super cyanobacterium that had a diversity of pigment types. It was involved in this primary endosymbiosis, which occurred just once, not multiple times independently, and then there was subsequent diversification into different lineages. This cyanobacteria, it was presumed, was swallowed by different eukaryotic lineages. Somewhat unsatisfactory as a hypothesis in the sense that you would have had loss in many different lineages of the different pigment types, because you don't find eukaryote chloroplasts that have the complete spectrum of pigments. They have red, they have green, they have brown. Right, so I began my studies as a PhD, and I have to tell you, I mean, I was attracted to this general problem, perhaps it had a lot to do with the opportunity to collect prochloron didemni. This is an organism, a photosynthetic bacterium that has the green pigments. It's found as a symbiont on the Great Barrier Reef. I used to collect it just at the reef crest here, and this was really what gave me the sort of the motivation to to undertake this study. All right, that's just some biological background, but stripped down to the question actually becomes, can we reconstruct the evolutionary history of chloroplasts from analyses of DNA and protein sequences? All right. To do that, we use substitution models. These provide a formal description of the process of sequence evolution. They're useful in a number of ways. We can use substitution models to, if we were to compare two sequences and we were to observe some differences between them, we can use these substitution models to estimate the total number of substitutions between two sequences that have diverged. This is very useful because it provides for distances that have nice properties for reconstructing evolutionary trees. If we have substitution models, we have a formal description of the way these sequences evolve. We can also calculate the probability that a given tree evolved the observed sequence data. I'm going to talk quite a bit about models. I want to get back to the chloroplast. So the different hypotheses of origin predict different tree topologies. We have some hypotheses of origin suggesting there was one primary event of endosymbiosis, and we have an alternative hypothesis that there are perhaps multiple events of primary endosymbiosis. And these are, in a sense, these are the sort of the, this was our naive thinking at the beginning of the project, that it was just simply a matter of collecting sequence data from different organisms, comparing the sequences, using a model-based approach to reliably reconstruct the evolutionary history and see which predictions were met. So, you know, very simply, what I tried to illustrate here, I've got a black circle which might represent non-photosynthetic bacteria. So we might imagine that the root of the tree joins the branch that leads to the non-photosynthetic bacteria. Then we might have the evolution of distinct cyanobacteria, and then we've had a primary event of endosymbiosis that's given rise to these plasters. If perhaps the present-day cyanobacteria have diversified themselves relatively recently, we might even find this kind of topology. If we had multiple events and a hypothesis, for example, that the present-day green cyanobacteria have given rise to the chloroplast in green plants, then perhaps we would see the sister relationship between those green cyanobacteria and the green chloroplast, and perhaps we'd see a sister relationship between the red chloroplast and the red cyanobacteria. Substitution models in common use. If you're a biologist, I think one of the best reviews of substitution models was written by Nick. How do you pronounce? Leo and Goldman. Okay. The principle here, in terms of a formal description of sequence evolution, is to describe the substitutions beside in terms of an instantaneous rate matrix, which has two parameters. A rate parameter concerning the substitution of one base type to another, a frequency parameter concerning the frequency of the bases, or in the case of amino acids, the equilibrium frequency of these in the sequences. These models, which are used, have three important properties. When we reconstruct evolutionary trees, we tend to assume that the same patterns of substitution are occurring on all of the lineages of the underlying phylogeny. We need to assume that the frequency of the bases or the amino acids are in equilibrium. We have this condition of time reversibility that's very helpful in simplifying calculations of the likelihood or the probability that a given tree evolved the data. It's just simply the proportion of the frequency of one particular residue times the instantaneous rate of substitution of that residue to another equals the proportion of that substituted residue times the instantaneous rate of substitution back to the other. If we develop such a model within this framework, it's possible to calculate the probability of change from any state to any other state along the branch of an evolutionary tree. If this is done, we have a means to calculate the objective distance or the total number of substitutions between sequences. We have a means to calculate, as I said, the likelihood that a particular tree evolved the sequence data. I want to talk about spatial heterogeneity because in itself, that as a description of sequence evolution is insufficient. We have the problem of spatial heterogeneity. And this is because sequences are not there for the benefit of molecular evolutionists to reconstruct evolutionary history. Sequences are constrained in a number of ways. Okay, typically the sequences we use to reconstruct the evolutionary history are part of enzyme complexes. there are important interactions between proteins in those complexes, and sometimes interactions between complexes. And these interactions mean that the pattern of substitution of what sites are actually able to accept substitutions, it's not simple. So in developing our model of substitution, we need some way of incorporating this complexity in the spatial pattern of substitution. And one of the earliest ways of doing this was suggested back in the 70s, where it was suggested that you could take account of this, this heterogeneity in the substitution process by assuming that there are discrete classes of rates. Like there are some sites in the sequences that are evolving at a slow rate, another set of sites that are evolving maybe at a faster rate, another set of sites at an even faster rate. It was realized you could describe such a set of rate classes using something like a gamma distribution, where you just needed a single parameter to describe the relative proportion of sites in the different rate classes. This, I think, perhaps the first implementation of this was by Zhang Yang, and shortly after we saw that this, of all the parameters that can be altered in terms of a substitution model, appeared to have the most significant impact on the trees that were reconstructed from sequences. It would change the topology. It would change the relative estimates of the branch lengths. So, and in many empirical examples, it seemed that this made a lot of sense. At the time that they put forward their model, there was an alternative model put forward to describe the spatial pattern of substitution. This was the model of Fitch and Markowicz. They studied cytochrome C sequences, and they had a very different model of spatial substitution. They thought that in any one of these cytochrome C sequences at a given point of time, perhaps only 10% of the sites were free to vary. Cytochrome C. And that they thought that over time, 10% of the sites remained variable, but which sites were variable actually changed over time. So you might have, for example, if you were to pick a site in a sequence alignment, you might have, and we've got two groups of two clades, you might have at this site in plants, this is invariable, whereas there's been some switch, and in this clade here, it's actually variable. So this was formally described by Mike Steele and Chris Tuffley in 1998. They had a simple model where they had a proportion of sites undergoing a covariant process where there was a switching rate from off to on. They specified a switching rate from on to off. It's a time reversible model, so the proportion of sites that are off times the switching rate of sites on equal the proportion of sites that are off times the switching rate from on to off. John Hulsenbeck took that a little bit further by just saying, well, so under the Tuffley and Steele model, when you were on at a particular rate. But Hulsenbeck took this further and said, well, when you were on, you could actually be at a different speed. And there was no way of switching between speeds, but you could switch from off to a different speed in the on category. There's another model developed by Nicolas Gaultier. He didn't allow switching off. He allowed switching between different speeds. And the number of categories here is, I didn't say that about the Hulsenbeck. He had a discrete number of classes that for rate, as did Nicolas Gaultier. Now, the discrete number of classes within a gamma distribution. All right. I've introduced the idea of there is spatial heterogeneity in sequences. It's been modeled successfully with a gamma distribution. There are other models to model spatial heterogeneity. And let's talk about now what happens if the model is misspecified when you're reconstructing a tree. It has two I like to think of it in terms of having two effects. It can reduce reconstruction accuracy. Okay, so what do I mean by that? Well, it's known somewhat problematic if the underlying topology of a tree is such that if you have long branches and long external branches, short internal branches, then it's quite difficult for tree reconstruction, to recover the correct tree. So Felsenstein first gave an example of this with a scenario where he suggested that there were fast rates of, what this diagram illustrates is fast rates of evolution in lineages A to D. And when you try and reconstruct the tree, you tend to reconstruct trees where A and D come together. He showed that this was particularly a problem for the method of parsimony. But in actual fact, if the substitution model is misspecified, then this is also a problem for model-based methods as well. This is just another example to show you can have, in Felsenstein's model, he imagined fast rates of change at given a site in a sequence alignment. He imagined that in some lineages the site was evolving fast, but at that site in other lineages it was evolving slowly. Penny and Hendy showed that the problem of this is what's called long branch attraction. The problem of long branch attraction isn't per se due to fast or slow rates. It's due to this juxtaposition of long and short edges. They showed a tree topology which could have arisen under a molecular clock where there is equal rates of change, but where there's short periods of divergence early on in the tree followed by long periods of divergence, then there's also difficulties for tree reconstruction. So you can have these difficult topologies for a number of different reasons. I'm going to give you a third in a minute. And if you've got model misspecification, then all methods have some difficulty here. An illustration of the problem with likelihood is made by Peter Waddell, where he showed, he did an analytical example where he showed that if you had a tree which had these extreme pendant or external branch lengths and short internal branch lengths, then in the presence of invariable sites in the sequence that can't change, and that's a reality with biological sequences. Many of the sites are constrained across all of the taxa, and these models have got to assume the sites are variable. So, in this example, Peter Waddell showed that if you had this extreme underlying topology, and you had sites in the sequences that were invariable, but you didn't take this into account, so the model was misspecified, then maximum likelihood of neighbour joining here tended to converge to the tree which favoured the long branch attraction, the coming together in this case of T1 and T3. So that's an example of how model misspecification can reduce accuracy. Here's another example, or this is another, not an example, this is another point. If the substitution model is misspecified, it can also induce, I don't know if it's the right word to use, but using the word topological distortion. If a substitution model is misspecified, it can induce those extreme topologies. Okay. Why might it do that? Well, probably the truth about sequence evolution is the process of sequence evolution is not expected to be a time reversible process. It doesn't look the same in different directions. I think there's been a number of biological observations to suggest this, but I think Hervé-Philippe's group really sum it up when they say in different lineages, coevolution of proteins canalizes the evolution of a protein in different directions. So as sequences evolve, they co-evolve with their partners. This is particularly true of proteins that interact with other proteins. The example that he liked to use was if you took rat cells, and if you took mice cells and you removed the mitochondria from them and then replaced them with rat mitochondria, even though the system could be sustained, the interactions between nuclear proteins and mitochondrial proteins was not sufficient so that the mitochondria could function. There's other examples of that. But what does it mean what does it mean in terms of substitution models and process of evolution if there are changes in protein-protein interactions? Well, again, Hervé-Philippe's group, they were thinking about this some time ago. the number of variable positions can be different between lineages, suggesting that a constancy is a limitation of the covariant model. So one thing about those covariant models, they assume that the proportion of sites of variable is the same in all the lineages. The thing is, though, if you relax the functional constraint of a protein in a particular lineage, you may well increase the proportion of sites that are variable. This was described more formally by Mike Steele and myself. Here's an example of a 4-taxon tree. I've got two models here. The first model is the model of Felsenstein, where Felsenstein put forward a model that said when the rates of evolution were faster in lineages B and C, might lead to a long branch attraction effect where B and C come together. Very similar results are obtained if you have a tree where you actually have a constant rate of evolution, but in lineage B and C, you increase the proportion of sites that are variable. And actually, you can see that as actually being, think of that in terms of being a mixture of trees, where there's a constant rate of evolution in all the lineages here, and a constant rate of evolution in the lineages that are variable here. It's just that there are only two that are variable, and you can get this problem of those branches coming together. We gave an example of this quite a long time ago in 1996, actually. An example where we showed that model misspecification had two effects: to reduce reconstruction accuracy, to induce topological distortion. It concerned a case and a study, I haven't got too much time to tell you about that, concerning the biosynthesis of chlorophyll and bacteria chlorophyll. And it concerned, it came down to an analysis of these homologs. They weren't actually orthologs, meaning they were not just related by divergence, they were also related by duplication. They were paralogs, some of these. So in organisms that carry out oxygenic photosynthesis, there's this gene, CHLL. There's a homologous gene, BCHLL, in organisms that carry out anoxygenic photosynthesis. There is a recently duplicated gene from BCHL. A question of interest was whether a sequence from an outgroup, which again had a different function, where did it join? Where did it join in this tree? And what we noticed was that there was in a sense, a topological distortion. Here, since BCHX had diverged from BCHL, it had increased in its proportion of variable sites, and so it was on a very long branch. If you wanted to test the placement of this gene in this tree, and you used a maximum likelihood and a method and a model where you assumed there was no invariable sites, you've got a very strong placement of this taxon on this edge. However, if you removed those invariable sites, then maximum likelihood could make a better estimation of the probabilities, and it recognized the length of this, and so it didn't give false support to this root placement. So this is just an example illustrating the impact of model misspecification in the two ways. I want to come on to heterotachy. There's been a lot of recent discussion about in the literature about this term heterotachy, and it's in some ways it has to do with spatial heterogeneity. The observation of heterotachy is summarized in this figure. Heterotachy concerns a site property of sequences. Here we've got some mammals. There's 4 clades of mammals, and these are observations about the number of substitutions that there are in the different clades at specific sites in the sequence. So, an illustration of that is that, actually I can illustrate with this. Is there a pointer? Oh, okay, that would be great. Sorry, I should have looked for that earlier on. Great. So at this site, there's no change in this group, but then there's change in this group. And then you might find other sites in the sequence where there's a lot of change in this group and none in this group. So heterotachy is the inference of a site property, that at a given site in a sequence alignment, there are different rates of evolution. There could be a number of different causes of heterotachy. This inference in itself doesn't tell you anything about the process of evolution that has given rise to it. And that's important because heterotachy has become a topic of conversation concerning the relative performance of different tree building methods. And the implication is that sequences have this complex pattern of spatial heterogeneity. It can be described by heterotachy. So let's simulate sequences under heterotachy and look at the relative performance of the methods. And the way that heterotachy is typically modeled is to, so if you don't know about simulations, given a tree and a substitution model, you can simulate sequences that fit that tree. So the way that heterotachy is modeled, and then once you've simulated those sequences, you can take those sequences and you can apply a tree reconstruction method to see if you can get back that model tree, the tree that you used to simulate the sequences. So here you go. So with heterotachy, what people do is they tend to take a tree, perhaps you don't have to worry about any of these numbers, but you can see the relative length of branches here. So A is long, the branch leading to A is long, the branch leading to C is long. And they will simulate sequences on this tree, a partition, so maybe 1,000 sites like that. And then they might simulate some other sequences where the tree is actually, it's the same tree, but the relative branch lengths are different. So here the branch length to A is short, branch length to B is long. And so they sequence a partition of sequences like this, and then they join them together, and then they try and reconstruct a tree with the different methods, assuming assuming in a sense that one set of branch lengths fits all the sites. And they've done this in different ways. So quite a number of people have simulated heterotachy in this way, where they've used different relative sizes of partitions, they've used different relative branch lengths. And then they've made statements about different methods of tree building. They've made statements about different methods of tree building as to how they perform. This is just to remind me that I don't understand, because the thing is that heterotachy was motivated by the inference that proportions of variable sites changed in different lineages. That doesn't have anything to, those simulations have nothing to do with that. So I'm not quite sure what they're modeling. All right, so the thing is that tree mixtures can also be used to simulate changing proportions of variable sites. Mike showed me how to do this. But I'm just going to show some results of some simulations where we have a tree, and what we're going to do is effectively in two non-adjacent lineages, we're going to increase the proportion of variable sites. So we're going to induce a topological distortion, and we're going to see what the reconstruction accuracy is. You know, are the methods going to fail to correctly place A and B as adjacent? Are they going to put these two what become long branches together, B and C? So here's a simple simulation where we've got 4 taxa. We're going to simulate a 4 taxon tree, and we've got the length of the branches are such that it's made to be a difficult problem. These are numbers of substitutions per site. 0.75 would be saturation, so they're quite long, 0.4. So I've got all of these external branches of 0.4 in length. Then I've got a short internal branch here. 1/3 of the way along this branch and this branch, I'm going to increase the proportion of variable sites. I'm going to start off the ancestral condition that there is only 20% of the sites in the sequences are variable. That's quite generous. Walter Fitch thought it was only 10%, but I've got giving it 20%. The sequences are going to be very long. I've sequenced these for 10,000 bases long. I'm going to, of the sites that are invariable, there's 80% of the sites are invariable. I'm going to first of all turn 2% of the sites on as making 2% variable, and 4% and 6%. and I'm going to turn on the same. I'm going to do it in a correlated way. So the sites that I'm turning on to be variable, I'm turning on the same sites in both B and C in this simulation. Okay, so this is how maximum likelihood method, maximum likelihood reconstruction performs. So what this shows is the yellow line is the correct tree. The blue line is the correct tree. The yellow line is identifies the tree where, oh, here it is up here. The yellow line identifies the tree where the long branch taxa come together, and that pink represents the other tree. And what we see is that by the time you've added in, you've turned 8% of sites on, made 8% of those sites, those invariable sites variable, there's low reconstruction accuracy. And this is in the case where my substitution model, I've just assumed 80% of the sites are invariable. If you were to estimate the proportion of sites that are invariable, this is the sort of results you get. Again, reconstruction accuracy falls when a relatively small number of sites are increased as being variable in the two lineages. This is quite an unusual result I don't really understand, but if you simulate sequences in this way, the way that I think sequences are more likely to be evolving, and you assume a discrete gamma distribution, then not only do you have poor reconstruction accuracy, you get quite strong support for the wrong tree. Interestingly, if you assume that not only in terms of the spatial heterogeneity, you assume not only that there's a discrete number of array classes of variable sites, there's also some sites that are off. You get low reconstruction accuracy, but no longer do you get strong support for the wrong tree. With the method of parsimony that makes no explicit assumption of a model, it probably performs the worst. We're going to round up and we're going to come back to the chloroplasts. I said at the beginning of the talk that when trees are built from different proteins, or what we hoped to do was, when I started my project, I went out on the Great Barrier Reef, I swam around, I collected some prochloron. It was pretty tough. And I took back to the lab and I sequenced some genes. And while I was doing that, the genome industry was, ploughing ahead, and there were a lot more trees generated over that period than I could generate. And if you look back over all the data and the data sets that have been collected, what you find is not trees that really fit either of our, don't really fit our simple predictions that we had at the start of the project, that might be simply interpreted in terms of, oh yes, there's been a single event of endosymbiosis, or there's been multiple events of endosymbiosis. What you see is actually a variety of different tree topologies. Some support a primary event of endosymbiosis, some support multiple events. So, what does that mean? Well, okay, so here's something. The thing is that an observation is that if you build trees for different proteins, if you build trees for proteins that are part of the same enzyme complexes, they're much more similar than the trees that are built for proteins that are parts of different enzyme complexes. And so what does this show? This shows, so I've got here 14 different proteins. And I've got, for each protein, there's the same 19 taxa. And for each of those proteins, I've calculated the support for different relationships, or splits, we might say splits, that are above a certain threshold. And then I've compared in a sense, the splits. So that's just the amount of support for different possible relationships for each of the different data sets. And so I've got just a Euclidean measure of distance, which is just measures whether a split's present in when you're comparing two two protein data sets where there are splits present, the relative support. So we've got a measure of distance between all possible spectra, you could call them, and then plotted them here in a PCA analysis. And what you see, for example, is the same color identifies. So there's 14. There's 14 proteins. There's two proteins in a particular enzyme complex. So, for example, the green are RPO-C1 and RPO-B from RNA polymerase complex. These are two ribosomal proteins. These are, which are not together. This is a little bit, I don't know if you can see that. These are part of the ATP synthetase. These are part of the electron transport chain. This one's quite different from this one. But what you don't see is you don't see these spectra centered around one place in this space here, which is kind of what you'd expect if there was maybe one true phylogeny and there was stochastic error around that. You would tend to see them centered, but you don't. You actually see a clustering of the in terms of their relationship to the same complex. The reason there's multiple spots for any given point is, so essentially what I've done is I've taken the protein so they're taking this protein alignment, and I've made an estimate of the variability by doing a resampling technique. So the scatter is sort of a measure of the variability in the estimation of this spectra. It's very non-random. All right, so questions that this leads to. Different trees, which suggest really quite different hypotheses of relationship, are obtained when you carry out phylogenetic analysis of proteins in chloroplasts and in cyanobacteria. Important question is whether these topological differences are caused by lineage-specific differences in structural and functional constraints. Is that the best way to explain these findings? And if so, it really motivates the next question, which is really an interest to develop low-parameter, non-time reversible models that would, that maybe would more accurately describe the evolution of the sequences. So, that would be really an important direction for research. I'm just going to finish now. So more than 5 years ago or so, someone said, we now have textbook-like certainty in our understanding of the early events of chloroplast evolution. And this really came about from those very first trees that were being generated. But then as more trees were generated, we realized that they were different. And so we still really don't have a very good understanding of the question that I first became interested in on the Barrier Reef 20 years ago. The thing is that there's still a lot of things to be done. There's important developments that might be made in model development to actually try and, and I don't want to be, I think the implementation of the gamma has really been, one of the most important things in model development, but, maybe we can do a little bit better than that. And so maybe there's still some more important things for mathematicians to do in terms of model development. Why I say 20 years on, there's still need for some more hunting. Well, there are many chloroplasts and organisms in these supergroups that have not been characterized at all. So we don't actually even have the empirical data for a lot of the groups that might be important in terms of the biological variation. So there's still need to actually maybe do some more diving on the Great Barrier Reef. All right, so we're pretty much at the end of the talk. I just wanted to thank. So this story today was really due to contributions from a number of people. So I've only given you part of the story in the sense that other biological data is also used to inform this question of chloroplast origins. And with Tony Larkham and Chris Howe, we've recently written a review of some of this information and talked a little bit about what I've said today in the context of the overall information. It's with Ellen Nisbet and Klaus Schlepp have been specifically looking at this phylogenetic spectra of the different photosynthetic proteins. And with Bill Martin and Nicole Grunheit, we've been doing analyses with different covariant models, and the simulations were also part of a study we were doing. Mike probably been involved really in the development of the ideas almost more than just about anybody else in terms of the models and that. And so it's important to thank Mike. And I want to thank very much all the organisers for today's symposium and generally for the opportunity to be here. And I got some money from Microsoft, so I won't say a bad word about them. Okay, so that's it. That's me done. Thanks very much.
You said more hunting might be needed. So, do you have any hope that this project of sequencing the whole ocean?
Actually, it actually could be a little bit more boring now, because there's a lot of interesting organisms in culture already, and we should just really go to the lab and work on those. So, yeah, I don't know what to expect from these surveys. I mean, I think they're important to do, but we'll see. So, if you don't know, John Cleese came to Palmerston North, and he gave a show. It didn't go down very well. And then he actually termed Palmerston North the most boring place in the universe. And then the council responded, which they had a good sense of humour, and they named a hill at the city dump after John Cleese. So, if you come to Palmerston North, this is one of the important things you must see. Okay.
Anyone else?
Right, could you back up? I'm just interested in the slide you showed. Yeah, I keep going on. That one. Could you just say a bit more about how you did that, please?
Yeah, so there's no model of correct. So p distances, a matrix of p distances. Then I've used distance Hadamard to calculate all splits above a given threshold. Then all of the splits in their weights have been compared to get a measure of distance, pairwise distance between all of the proteins. Okay, and then those distances have been, the relative distances have been displayed in a PCA analysis. You could try, it'd be interesting, you could try different models of correction and look to it. I only got this yesterday actually, so I haven't taken this too much further.
Talking about something very similar with students.
Oh, okay.
For lunch.
Oh, okay.
And we concluded that no one had done anything like that.
Oh, so well, I only got this one figure yesterday.
So you win by 12 hours.
Yeah.
I'll talk about that.
Okay, sure.
Actually, I have a question.
Yeah.
When you were showing the slides with increased proportion of variable sites,
Yeah.
what model were the variable sites?
So that was just Duke's Cantor model.
It was Duke's
Yeah, so I just kept it really simple. Oh, actually, so I beg your pardon. So I've done it with just Duke's Cantor, and these are actually the variable sites of Duke's Cantor, but I've also got a Tuffley Steel process ticking in the background here, because I thought that was maybe I capture what I thought was the essence of sequence evolution. But even without that, with just Duke's Cantor, and when I reconstructed, I always just used Duke's Cantor. So when but I get similar results whether I leave the Tuffley and Steel out altogether. So.
In these diagrams which just follow, it was very clear that sort of course it's due to construction, one of the three splits never shows up.
Yeah.
And of course it's also a wrong split. The other two don't know so much. So this seems to be an argument in favor of split decomposition theory, where you say, okay, I look for those splits that are supported, even on the quadruple side, even if they are incompatible ones.
Yeah.
You try to fix that into something tree-like.
Yeah.
Would you also?
Yeah, that's right, because there's never any support for the third possibility, and you'd see the support for these two. I mean, yeah.
So as you don't know, I mean, here you know what is true and what is not
Yeah.
true, but in reality,
Yeah. What?
you
So
just see
Yeah.
two quartet splits have some support, and a fourth one doesn't. Then at least you have some information that you should try to use.
Yep.
Great. Let's thank Pete again.
I'll just get a glass of water. Oh.
I know, I heard.
Sorry.
I didn't want to interrupt you.
Great. I'll just take that. I'll just take my seat. Thank you. Oh yeah. Oh, were you? Oh, I'm sorry. I was miles away.
Great.
Is Taylor?
She should be around.